Dr. Becnel served as the Assistant Professor of the Baylor College of Medicine, Departments of Medicine, Hematology, Oncology Section, and Molecular and Cellular Biology. She served as the Director of the Dan L. Duncan Comprehensive Cancer Center's Biomedical Informatics Group and Co-Director of the Biostatistics and Informatics Shared Resources. She leads the steering committee of the Collaborative Bridge Translational Research Domain Analysis Model, and this harmonizes the CDISC foundational standards. In addition, Dr. Becnel has served as principal investigator and IT director on dozens of grants and federal contracts, including national clinical trials, internationally utilized basic sciences databases, national data sharing initiatives, as well as some national and statewide cancer biobanks. Today, she will provide an overview of CDISC clinical protocol, analysis, design, therapeutic area, genetic and other standards, as well as the controlled terminologies and other semantics to support them with real-world use cases of how implementation of these standards can help uncover new discoveries. She will also describe SHARE and how it can be used by the academic community. I want to thank her for her partnership in communicating this information to us today. Before I pass the control of the slides over to Dr. Becnel, who will begin today's talk, I want to remind you all to put yourselves on mute to help avoid some of the background noises. We want to hear from all of you, all of your questions and comments, but until the end. And please note that this session is being recorded and will be made available for future use on our portal, BD2KBiostandards.org. At this point, I'm going to hand it over to Dr. Becnel. If I can. Lauren, do you want me to just pass the ball to you, or do you want me to work the slides?
Oh, sure, you can pass control over to me.
All right, I just have to find you. There you are. Do you have it?
It's not alerting me. I don't think I do, Rhonda. Okay, maybe it would be better for the sake of time to just get started and I can ask you to go to the next slide.
Sure, sounds good. Can everybody see the first slide? Yes. Yep. Okay, great.
Well, apologies for the technical difficulties in being able to reach you all today, and thank you for joining the call. It's my pleasure to give an overview of the standards that have been developed for regulated clinical and translational research by the Clinical Data Interchange Standards Consortium, or CDISC. Next slide, please. CDISC was established almost 14 or 16 years ago, rather, to be a worldwide standards development organization, or SDO. They recognized a gap in the clinical research space, such that there really weren't a lot of standards to support regulated research protocols for submission to FDA, EMA within the EU, PMDA within Japan, and so forth. So Dr. Rebecca Cush and Bron Kisler founded CDISC to support the creation of standards for electronic acquisition, exchange, submission, and archiving of clinical research data and metadata. We now are, 16 years later, a consensus-driven, community member-filled organization. So our standards are created by largely volunteer teams who have some CDISC staff to help support them. These standards are all open and freely available on the website at www.cdisc.org. All you have to do is click under the Standards tab at the top, and you'll be able to access them. Next slide, please. So as I mentioned, we were founded in '97 and incorporated in 2000. CDISC now has just over 400 organizational members. This membership is quite diverse. The bulk of our members are from biopharma corporations, but there are also members from academia, governmental bodies, service and technology providers, and others. These members, as well as other participants from the regulated research community, all come together to support the creation of a variety of standards about which I'll tell you today. These standards are in use worldwide. So the Americas, Southeast Asia, the Indian subcontinent, and Europe have all taken advantage of CDISC standards, participation, training, and these standards have been downloaded in more than 90 countries total. And to the right here, you can see a little snapshot of the CDISC website. Next slide, please. So why do we develop standards? Well, of course, as many of us know, metadata and data standards help support semantic and syntactic interoperability, which is to say meaningful data exchange. It allows us to aggregate data from lots of different groups and to be able to analyze them and come to meaningful conclusions. But that sort of research goal isn't the only driver. Another reason why we're interested in standards is strictly a business case, cost and time savings. So in this particular slide, I'm showing you at the top left, the kind of protocol creation steps that any clinical protocol has to undergo. These protocols then have to be IRB approved. Going more towards the right, you have patient enrollment. At some point, especially for interventional clinical trials, you lock your database to perform analysis, and then you do a final report out. If this trial is an IND or IDE for a new drug or device, or another trial that is regulated under the mandates of FDA here in the US, you then have to prepare your data for submission at bottom left. CDISC partnered with Gartner's a few years ago to assess what is the total time and monetary savings of standards, because as we know, to implement standards is not free up front. You do have to put in sink some cost into it. But once that's done, what do you get from it? What we found is a 20 to 30% savings for the following assumptions that are here at the bottom in purple, which for groups that are doing a high throughput volume of trials, that can be anywhere between $40,000 to about $250,000. Next slide, please. So we've established this case of who we are and why we're doing what we're doing, but in a more broad sense, we are helping to support precision medicine and a learning health system. And if those of you on the call are not familiar with the learning health system, that is, in a nutshell, this ability to have clinical and translational research take place in an environment where it is embedded within electronic health systems and with health providers, and where what we find in research can inform what's done in clinic and vice versa. I'm sure we're all already familiar with the President's Precision Medicine Initiative, which various institutes within the National Institutes of Health are participating in. And so on the next slide, I'll tell you a little bit about how we at CDISC have had very important collaborations over the years with a number of different biomedical domains as evidenced by partnerships with different institutes within the NIH. So at top left, the National Cancer Institute is by far our deepest, broadest, and longest standing partner. They have assisted us in thinking about how we manage our controlled terminologies, and as I'll tell you in a moment, we have an extremely close partnership with them on controlled terminologies, which I may refer to sometimes as CTs. We also have worked with them to co-develop as stakeholders the bridge domain analysis model, which you'll hear about more in a minute. And then we've worked with them on share metadata. And share is the metadata repository that CDISC has created that Rhonda mentioned in her introduction. At top right, we've done some work with NINDS to pull their common data elements, or CDEs, to use them for Parkinson's and traumatic brain injury therapeutic areas, or TAs, that are used to support FDA submissions. So with NIAID, they've actually adopted CDISC standards for FDA submissions. They're also using these standards for pharmacovigilance studies and for meta-analyses from many different funded investigators under various NIAID programs. For mental health with NIMH, we have used their CDEs to contribute to a schizophrenia therapeutic area standard. And we are about to work with them on a post-traumatic stress therapeutic area standard using their CDEs. In BD2K and NACHD, we've worked with both of them as well to develop therapeutic areas for the former and pediatric terminologies with additional support from NCI's Enterprise Vocabulary Services, or EVS team. And I'll tell you a little bit more about the BD2K example with a specific real-world use case of how these standards can actually start to help both clinical researchers and patients. Next slide, please. But the support of precision medicine long-term, learning health systems, isn't just limited to our research centers at NIH, but also to global regulatory bodies. So here I'm showing you a variety of documents that were released by the FDA in December 2014. Perhaps most importantly is a take-home message from this, or our talk today, that beginning in December 2016, FDA will require that all submissions from new protocols conform to SDTM, our Study Data Tabulation Model, and SEND, which is Electronic Non-Clinical Data, standards. If you have no idea what those things are, don't worry about it. I'll tell you more about those standards from CDISC in a moment. But suffice to say, suddenly any pharma, cooperative group, or investigator-initiated protocol that will be regulated by FDA must begin to use CDISC standards in 2016. Next slide, please. And you'll note that I mentioned global, so it's not just FDA. So Japan's FDA counterpart, the PMDA, will also require use of those same CDISC standards beginning in December 2016. And this is important for global trials that span US and Japan. Next slide, please. So now that I've given you a bit of introduction for CDISC and kind of the generalities, I'd like to transition to describing what standards we have that are available, how you can access them, and in the end, how you can get involved in some of the activities that we have, since we are a community-driven, consensus-based SDO. So first, I'm going to drill down on what we call CDISC's foundational standards. Next slide, please. So at the top, there's a green arrow that kind of takes us through that planning, collections, through reporting phase, and that's pretty similar to what I showed you for our business case before. Just underneath that, there are a variety of artifacts that one creates over the course of a trial. They range from an actual protocol document that has to be submitted to the IRB for approval, case report forms, which are instantiated in electronic data capture systems as electronic CRFs. You have data management processes, analysis and reports, and then finally you archive the data for posterity and for submission to clinicaltrials.gov, WHO, and others. So we have developed a variety of standards that really support every activity and every step of this life cycle. These standards tend to be very general, so they can support any clinical protocol, but can be especially used for regulated protocols. So at the level of the case report form, there's something that's called CDISC, for Clinical Data Acquisition Standards Harmonization. CDISC provides a snapshot view of SDTM, which I'll get to in a moment, and it really gives you the 200 most important data elements that you would find in almost any case report form. So things like patient demographics, for example, would be included there, and these can be reused. For SDTM, which I've mentioned a couple of times, this is a study data tabulation model where you have metadata elements that give you standards for capturing and managing, and even to a certain extent reporting data for clinical studies, and also specialty areas such as medical devices, pharmacogenomics, and questionnaires. To the right, for reporting and analysis, there is an analysis data model called ADAM. And this is something where you kind of summarize your design and your analysis plan for FDA to show them what you're doing. And then to the bottom left There is SEND, which is a standard for exchange of non-clinical data. So you may be thinking, non-clinical data, what does that have to do with a clinical trial? Well, this is where you capture information on co-clinical studies, for example, with mice or other animals that you've used as a model organism. Underlying all of this in purple at the bottom are CDISC control terminologies. Next slide, please. So to focus on SDTM for a moment, this is the kind of a visual representation of SDTM's implementation guide, or IG. And you'll see these different colored elements are classes or broad categories of information that one has to collect for clinical trials. Each of the smaller slices in those colors are domains under which there can be multiple different variables that one can collect for a trial. SDTM incorporates all of these, which means that if you are a clinical informatics support person at your institution, you and your investigators don't have to reinvent the wheel. Many of the metadata for data elements in your CRFs that you'd like to collect already exist and have been generated by the community through consensus. Next slide, please. So previously I mentioned control terminologies and how deeply important they are for essentially all of CDISC standards. These were not developed by CDISC alone. So when control terminologies first started to be developed back in the early 2000s, CDISC realized that NCI's EVS team already had dedicated terminology and biocuration experts. They had an extremely robust infrastructure. They had a variety of standard operating procedures and other resources. So we partnered with them to leverage this real strength that they had. And in fact, if you want to today suggest a new term for a CDISC control terminology, you wouldn't do this on the CDISC site. You would use NCI's term suggestion tool. So that shows how deep that relationship is. So now all of our CDISC terminologies are available via cancer.gov, and we are in this joint collaborative effort using EVS infrastructure to develop, maintain, publish, and version all of our standards for these terminologies. It's important also to note that all CDISC terms have an NCI C code. And if you've ever used the thesaurus or any of the other EVS products, you'll know that with that C code, you can link to other terminology sets because NCI is not just partnering with CDISC. They have many other community partners. And so now this lets you kind of walk across from CDISC standard terminologies to others. Next slide, please. So I mentioned that we started creating these control terminologies with NCI in 2006, and at that time there were about just over 600 terms. As of 2016, I think that we've gotten over 30,000. And although this number has been growing steadily, suffice to say, these are not duplicated terms. There's careful human curation, and so these are standards that have been used by the community and that are very high quality, but also very comprehensive at this point. Next slide, please. So now that you have some understanding of the foundational standards from CDISC, if you weren't already familiar with some of them, I'd like to tell you a little bit about a domain analysis model called BRIDGE, which stands for the Biomedical Research Integrated Domain Group. Next slide, please. If you've never heard of a domain analysis model, don't worry about it. You can see a picture at the bottom, and this is not intended to be a visual acuity test. I know that no one can read any of these things, but what this shows you is that the domain analysis model, or DAM, is a very large UML, or a type of unified modeling language, diagram that visualizes many of those foundational standards from CDISC that I mentioned, as well as standards that were developed from academic stakeholders, the NCI, FDA, and others. BRIDGE is now an HL7 and an ISO, or International Standards Organization, standard. This DAM can be used to create software. So even though it's UML, it's something that programmers can use to help create and stub out code. And it's written in such a way that the examples and other definitions are readable and interpretable by subject matter experts. So it bridges that gap between technical and subject matter expertise. It is, so far as I know, the only model that cross-cuts biology and clinical research. So up until about 1.5 years ago, Bridge really focused on clinical research. So it had concepts around adverse events and other clinical activities. But recently, it was harmonized with NCI's Life Sciences Domain Analysis Model, or LSDM, and it subsumed that model. And so now it really spans the entire breadth of biomedical sciences to include biospecimens and molecular biology terms. What's important to note is that it also implements healthcare data types. If you're familiar with ISO 21090 data types, that's what Bridge uses. And so it's also a hook for us to get into healthcare in support of a learning health system. Next slide, please. So Bridge can be used to visualize some of the foundational standards. And now I'm going to tell you about therapeutic area and indication standards, which are layered on top of the foundational standards. So if you're not familiar with TAs and indications, within oncology, for example, that would be our therapeutic area, and breast cancer, prostate cancer, lung cancer, those would be specific indications. Next slide, please. So CFAST, or the Coalition for Accelerating Standards and Therapies, is an initiative that CDISC and its partner, the Critical Path Institute, founded to help essentially create and maintain therapeutic area standards that can be used to promote public health. Next slide, please. As part of CFAST, there are 2 committees. One is on the left, a scientific advisory committee that consists of the founding members, plus FDA, NIH, Transcelerate Biopharma, the EU Innovative Medicines Initiative, and ACRO. On the right, there is a steering committee that has largely the same membership, except that here there are members of the National Cancer Institute's EVS team. And you can see a bit about how these 2 groups work together to provide support to the community and CDISC staff teams that are creating these content in some cases. In other cases, CPAP is taking the lead on creating some of these TAs. Next slide, please. So I mentioned foundational standards and how TAs layer on top of them. And what I mean by that is at the bottom you see CFAST TA products. And all of the concepts and variables within a TA actually map back to SDTM, map back to SEND, map back to ATOM. And this is to provide traceability from general clinical research concepts to now, how are those research concepts implemented specifically and defined? What are examples for them specifically within a particular therapeutic area or given indication? Next slide, please. All of these therapeutic area user guides contain biomedical concepts. And what I mean by that is really illustrated quite nicely in this concept map, which is developed by a product that's been put out by a group in Florida. It's a free and open tool called CMAP. And in it, you see that there are a variety of entities, such as subject in blue in the top left. You've got biospecimen in green, tissue specimen in baby blue. And these biomedical concepts take the metadata from a therapeutic area standard and define what are the relationships between and among those concepts. It also can give you specific subsets of control terminologies. So you might not want every anatomic site for a biopsy if you're dealing with breast. You might want breast-specific sites, for example. It also can tell you some validation rules and other information that's really critical when you're thinking about implementing this in an automated electronic fashion for clinical research in systems from different organizations worldwide. Next slide, please. So CFAST has developed dozens of therapeutic area standards, and you can see the pipeline that we have here from things that were developed prior to CFAST being initiated to things that are in the till for 2016. One of the things that you'll notice is that there are a variety of sponsors for this that include FDA, various institutes from the NIH, as well as patient advocacy groups. There also are a variety of therapeutic areas that map back to different institutes. So for example, for NIAID, we have virology, tuberculosis, malaria, vaccines. For NCI, there's breast and prostate cancer. Others are coming. For NINDS, we've got Parkinson's and Alzheimer's. And so this is not something that's just limited to one particular disease area or disease focus, but rather it's very broad to support precision medicine long-term goals. One thing to note here is that the dyslipidemia and chronic hepatitis C virus, TAs, were mentioned specifically within FDA's technical conformance guide that I showed you on that slide previously. These are not required, but they are considered, they're included in the guide for consideration. Next slide, please. So we have a variety of standards for general clinical research, visualizing and developing software from Bridge, and now with therapeutic areas of standardizing not just only on the generalities of trials, but now having multiple different trials in the same disease area using the same standards that we can have meta-analyses for data aggregation. Historically, however, all of the content of these standards has been released via PDF. That's great because we can freely share them on our website. It's easy for us as human beings to understand them. But the drawback there is that PDFs are not really machine readable. There certainly are programs that can pull text out of PDFs, but it's not structured. Those programs tend to be not very accurate, and we lose a lot of the important metadata from that process. Next slide, please. So in response to this, CDISC created SHARE, which is a metadata repository. If you're not familiar with the metadata repository, or MDR, that's really just a database of metadata, or in this case, a database of all the standards that I told you about previously. So at the bottom, there is a variety of standards from CDISC that are versioned and controlled. These can be semantics and other standards. These are loaded into CDISC SHARE, and then there are a variety of tools around that to support quality control, making sure that there's good quality assurance and biocuration, so that there's quality product within the MDR. SHARE can then be used by a variety of groups. So we have human beings, such as programmers, biostatisticians, and data managers. External MDRs can consume the library that's within SHARE wholesale, so you can have your own CDISC library set up within your MDR or within your electronic data capture system. There are a variety of clinical research applications that can use this. So, for example, in some organizations, all clinical research management occurs within a single system. In others, they might separate protocol management from electronic data capture, from analysis, and other tasks. And SHARE can feed all of those. As part of a learning health system, SHARE can be used to feed data into clinical care apps or into applications that bring together information from electronic health records, wearables, and other systems with clinical research standards and electronic systems. But our bottom line, of course, is to help researchers so that patients can derive benefit from all of this. So in short, SHARE provides access to human-curated, computable CDISC standards. That helps the standards development volunteers and CDISC staff to support reuse of metadata concepts that have already been created, to manage those metadata standards. It helps people who are end users of our content to implement standards in real time, to meaningfully exchange research data and health data. Over time, it can reduce cost, as I showed you in the business case, and it will help you maintain compliance with a variety of regulatory bodies worldwide. So recently at the European Interchange, a variety of biopharma companies and Danone, who makes yogurt and other nutraceuticals, shared their metadata repository strategy, and in each they had some plan for including share within their MDR. So I think for a lot of industry-sponsored studies, this is coming, and so you'll be seeing those in those studies. It's important to note here that share exports are free and open to academics via eShare. We're in the process of updating our website to help support this request, but for now you can just shoot an email to me and I can make those things available to you until the website is updated. But these will always be free and open. Next slide, please. So at the bottom, there are a variety of different web clients, and these are the different types of apps and people that I told you about that will consume information from Share. But at the top of this particular graph, there's the Share MDR itself. There is a metadata service bus that we use to load data in. Our human curators are touching that within Share. On the right, there is eShare. So this bus is used to export content, and you'll see eShare in a moment. And then finally, there's the Share API. So we do have a first version of this that's undergoing internal user acceptance testing. We will be making this API public later in the year. And because it is not free to us to host or develop either Share or its API, we are going to have a business model around that. We anticipate that that will be out later this year. Although eShare will always be free, the API may not be free to some individuals, and more to come on that in the future. Next slide. So why is Share important? You know, we've talked about electronic access, we've talked about not limiting yourself to PDFs, but Peter Van Russel from Business and Decision put together this slide in January 2016 that shows a variety of foundational standards in blue, technical standards, different flavors of XML, semantics like bridge or control terminologies, and TAs in pink. You'll see that there's an increasing number from 2002 to 2010, next slide, and that this has continued to increase over time, such that not only have we had new therapeutic areas coming on board, but we have continued to work with our volunteers to version various other foundational standards. It is really not possible with PDFs to cross-compare different releases of a particular standard, or to say, if I have a concept that's used in 5 different standards, can I easily make sure that it's given the same examples were appropriate, that it has the same control terminologies, that that consistency is there. Share provides this. So it's quite powerful when you're talking about, you know, this dynamic standards body. One important thing to note here is that this line is not going to continue to go up ad infinitum. We have heard from our member community to slow down a bit, even though there's a lot of enthusiasm around development of new standards because they're high quality. It's hard for people to implement those very rapidly. So we have heard that. The other thing is that it's not that there will be an infinite number of therapeutic areas. FDA has identified 66 of these, and we have made substantial progress. I think up to 70% have some work that's been completed on them. So this will normalize at some point, although there will be continued maintenance over time for the core standards. Next slide, please. So I mentioned eShare previously, and there is a snapshot of eShare from the members only section of the website. If your organization is a member, you can log in using your email address for your organization and have access to a wide variety of content, including eShare downloads. It has CDISC, SDTM, ADAM, SEND, and all of the other foundational standards and derivatives that I told you about. It's got therapeutic area normative content, mappings between things, some control terminologies in the form of packages of control terms, although for now, if you want a whole listing, definitely go to cancer.gov and then diff files. And one important thing to note on the right is that for each of these different standards, there are a variety of different file types that you can use. These range from Excel to different flavors of XML, including ODM, which is really popular for academics, and RDF, which actually has an information model built into the metadata. So again, these are free upon request to members of the academic community if your organization is not already a member. Next slide. And finally, SHARE is going to participate as part of this learning health system. Dr. Michael Ibarra is head of Healthcare Link within CDISC, and he is started this project called eSource that's engaging 6 different task forces within the community to think through how we might be able to take data from the EHR, shown here in the middle, pull out a subset of that data that's important for a clinical research study and pull it into a researcher's data system, transform those data a little bit because maybe you don't care about all the information or you need to have the data aligned to appropriate structures for submission, and then submit those processed data or massaged data to the appropriate regulatory agency along with an approved audit trail. So there's a lot of work to be done in this area. There's a lot of community involvement. We're trying to align with things that the Office of the National Coordinator and others are doing in this space. And SHARE will play an important part of distributing clinical research standards to ensure regulatory compliance for this process. And finally, we are looking into having some syntax support for SHARE in the form of FHIR. Many of you might already be familiar with HL7's RESTful resources or REST services that they've developed within FHIR. It's a set of, each resource has 20 to 40 elements that are generally useful for health care. So this can be imaging, for example, patients, and other really standard health sorts of assets. So you're able to take an existing resource and extend it to meet a particular need, and that's called a profile. And so in the future, we might have our biomedical concepts or our foundational standards, for example, be represented via SHARE as CDISC profiles as part of SHARE on FHIR. Next slide, please. So now that you've heard a bit about our standards, our metadata repository, I'd like to give you a real-world use case for how CDISC standards can help make new discoveries and eventually impact the lives of patients. Next slide. So in this case, we're considering polycystic kidney disease, or PKD, as a use case. So on the x-axis, you can see age as a function of years. And there are a variety of images of a kidney as a patient progresses from a relatively normal kidney to an increasing number of cysts, and finally cysts that really essentially take over the kidney such that kidney function on the y-axis drops precipitously. When a patient has just started to have cysts form, they may not be experiencing symptoms. So if you think to look, there may be protein in the urine or more concentrated urine. There also might be hypertension, although we know that that's kind of deemed a silent killer because it's so frequently asymptomatic. As things progress, patients might start to experience pain, blood in their urine, infections, and other side effects of PKD. But really right now, the only clinical endpoint that we have is that drop of kidney function. And by then it's difficult to intervene, and for some patients it may even be too late to intervene. So where we want to get is to find a biomarker that will identify this disease early so that we can have early interventions to prevent progression or to even slow it if we can't prevent it. Next slide, please. So in this PKD project, the PKD Foundation, with support from Tufts University, CDISC, and CPATH, came together to say, look, we have a lot of extant data from multiple longitudinal, well-executed studies that are in a variety of different databases and registries. Those data are not standardized, so we can't do meta-analyses. let's come up with a PKD therapeutic area standard. Let's map all of the existing data to that standard, and then let's apply the TA standard to prospective studies. So now we have all of the data in alignment that we can analyze. Next slide, please. This actually has happened, and the outcome was that total kidney volume, or TKV, was discovered as a biomarker. It's not something new. TKV had been there in the data the entire time. However, because we were not able to do a meta-analysis across all of these different studies, it effectively remained hidden. It was found to be highly sensitive and specific, and it could predict progression not only of PKD, but of end-stage renal failure, which is sometimes called ADPKD. Dr. Ron Perrone has submitted an application to both the FDA and the EMA, which is FDA's counterpart in the European Union, to qualify total kidney volume as being, quote, fit for use. So this means that you can use this as a biomarker to evaluate whether or not a new treatment is actually efficacious for polycystic kidney disease. And so, in summary, the adoption of this biomarker will speed development of new clinical therapies and interventions to slow or stop the progression to advanced kidney disease and even renal failure. Next slide. So I covered a lot of ground today, and I really implore you not only to ask me questions today and to reach out to me. My email address is here. I'm happy to discuss any portions that we can't get to in questions, but also to check out our website. It is being updated now, but you can certainly log on to get copies of our standards in PDF. You can find ways to volunteer in any of the teams that I mentioned today. If you are a clinical informaticist who has to support a clinical trials unit within your organization and you're thinking, I have a variety of investigator-initiated INDs or IDEs that I need to support, I'd like to learn more, we have a variety of educational materials on the website, and we offer the opportunity to learn about CDISC standards from CDISC, both online and in face-to-face trainings that take place all over the world. You can sign up for news and newsletters, email blasts, see upcoming meetings such as the International Interchange, which will take place here in the United States in September, and find other resources. So with that, I thank you for your attention, and I'd like to welcome any questions.
This is Mike Herta speaking. I've got a question about what is the, I guess the basic question is, if we've got investigators at NIH and they're interested in using your standards and your CDEs and so forth, what is the cost? How do they do that? You know, what are the basic points there?
Great question. So in terms of cost, we don't charge anyone to use our standards. They can download them right now and start implementing them in I guess the least expensive way, of course, would be to download CDASH, and because that's part of SDTM, and to start using those data elements within case report forms. For larger compliance with FDA, I would say the best thing to do is to get training in SDTM and SEND. These are very mature standards, so there's a lot of different component parts to them, and I think investigators will find it much more easy to use after they've got a little bit of education. I think the standards in terms of a point of entry, the barrier is low. It's just when you start to implement them, you get into some specific questions. Without training, you can get a little stuck. But the total cost of ownership kind of depends on the systems that are in place. If your investigators are using a clinical research management system like Medidata Raise, then they'd want to create a library of metadata if your system allows that, or if it only thinks in terms of case report forms, create a library of case report forms that incorporate these standards. Does that answer your question?
Yep, thank you.
This is Bob Clark. I have a related question. So To what extent do the current commercially available clinical trial management systems actually include CDISC as a part of their package?
Great question, Bob. So there is some variation. I think some vendors have a bit more inclusion of CDISC than others, but for the most part, there's not much yet. I think there are some EDC products that have been used for cooperative group studies and for pharma-sponsored studies that have more CDISC standards baked in. But my prediction would be that with this coming mandate for regulated trials, that we're going to see more of that over the next couple of years.
I would hope so.
Yes.
Thank you.
Are there any other questions for Dr. Becnel? Well, I want to thank you, Dr. Becnel, for totally interesting, fascinating, thorough, and comprehensive webinar today. That's pretty much all the time we have left for today. On behalf of Dr. Becnel and the team here at the BD2K Standards Coordinating Center, I thank everyone for joining us and for your participation in this presentation. Have a great afternoon, everyone. Thank you.